Welcome, everyone. I'm Joanne Geer. We'll get started in just a few minutes. The Biopharma Research Council, we want to welcome you here today. This series was developed through the efforts of all of the speakers and the key organizer, Winston Kuo, who you'll hear a little bit more about in a moment. But we really appreciate the way experts with knowledge us develop these wonderful educational programs. We're going to do a very short poll and ask you just one question about yourself. Here we go. So while you're doing that, I'll just mention a couple of things. The slides and the recording will be available online afterward. We'll be sending out an email to let everyone know with the links and so on. And the other important thing is that on the right-hand side of your screen, you have a section called questions, and that's the perfect place to enter questions. We'll be watching. You can also put if you have technical issues there. We'll keep an eye on that as well and do our best to help you. But typically things have gone pretty smoothly here. All right, just another 10 seconds on the poll. Looks like maybe we have our results. Great. Oh, beautiful. And here are the results. Good. Okay. So now I'm going to close. So it looks like everyone's got either a significant active interest in exosomes or getting interested. So that's terrific. I'm going to hide that. And now you should see my slide. I'm just going to tell you a little bit about our upcoming programs. In October, in North Carolina, we do a one-day program that's all about just having people come together to share the work that they're doing. Virtually all the presentations are going to talk about collaborations, how they happen, what the scientific progress was, and so on. We do webinars on a regular basis. We have a series called The Mobile Patient, and in November we're doing a webinar on FDA guidance related to devices intended for the home. In March, we're doing an executive retreat program. It's really wonderful. It's the 6th time we're doing it, and it's really about bringing people together who are suppliers to industry in preclinical and clinical services and their counterparts in pharma and biotech to really talk through, how can we, what are some best practices, essentially, as it sounds, in creating partnerships, developing contracts, and so on, and lots of great examples. And it's a nice small meeting, usually less than 100 people, so it's a nice intimate 3-day session. I just wanted to underscore again, if you have any questions, enter them at any time. After our presenter, we'll have enough time to handle a number of questions and discussions. We just want to thank everyone that's been involved with this, and here we go. I want to introduce to you Winston Kuo, who put together the whole series. Winston is the COO of IES Diagnostics, which is developing diagnostics based on a technology licensed from the FDA, focusing on systemic lupus erythematosus. He's also editor-in-chief and founder of the Journal of Circulating Biomarkers. And he sits on the advisory board of Exosomics and Hansa Biomed. He's focused, which are focused on exosome-based diagnostics and life science products, respectively. His research interest in extracellular vesicles includes the technical aspect of commercializing both from life science and clinical aspects. And I'm just going to quickly go through, I know I introduced you too soon, Winston. I'm just going to share with you the other sessions we've had, which these previous sessions are already on the website. We had Jan Latval from Sweden. We had Johan Skog from Exosome Diagnostics. Today we're hearing from Sasha Vlasov. Next week we'll be hearing from Angel Ayuso-Sacido and Eva Rode on the regulatory and funding issues. Winston, I'm going to set you up right now. Would you like to go ahead and introduce yourself anymore and introduce Sasha?
Sure. Thank you, Joanne. Hello, everyone. I'm very delighted in collaboration with the Biopharma Research Council and Intech Publishers to provide you the short course in an exciting field of extracellular vesicles. And this theme is the transition from tissue to liquid biopsies. And as Joanne had mentioned, we had, Dr. Jan 2 weeks ago and Dr. Johan Skog's talk last week. They both gave outstanding and informative presentations. Jan gave it on an overview of the field, and Johan gave it on pretty much the clinical applications. And you can access their presentation talks using the link on the Biopharma Research Council site under the short course in extracellular vesicles. So today we have the pleasure to have Dr. Sasha Vasilov. We'll discuss in this session the commercial areas as it relates to Thermo Fisher's pipeline in the extracellular vesicles ecosystem, including those areas that we really don't associate extracellular vesicles with. So now we know why this is such an important field. Extracellular vesicles encompassing both exosomes and microvesicles. We know that they carry a variety of biomacromolecules ranging from nucleic acids to lipids to proteins. And within the past 5 years, as you heard from both Jan and Johan, that they have emerged as a promising, minimally invasive, novel source of material for diagnostics. So, these can be identified and collected from biological fluids. Of course, further research and proper tools, like what Sasha will be discussing today, is needed to make them more useful in the clinical setting. So we will hear areas today in reference to the commercialization aspect. So there are two more live sessions, including one today, and next week, we will discuss the regulatory aspect and information reference to potential funding opportunities. So let me pass this to Dr. Sasha Vasilov. I'm going to make an introduction first in terms of he's a developer and an application specialist with a vision who is currently at Thermo Fisher developing and providing tools to facilitate our extracellular vesicle research efforts. He's a senior scientist, group leader at Thermo Fisher. Sasha has developed multiple product lines, including reagents for exosome research, siRNA, microRNA, mimics and inhibitors, and transfection reagents. Currently, he's focusing on developing of advanced technologies for exosome analysis and improving methods for extracting nucleic acids from clinically relevant samples. Prior to joining Thermo Fisher, which he was at Life Technologies, and he was a senior scientist at Somogenics, postdoctoral fellow at UC Boulder in Colorado, a visiting fellow in IBMC, France, and a research scientist in Russia. And during his scientific career, Dr. Vasilov co-authored over 70 publications, has currently 8 patents. So just a few housekeeping items before we begin. Sasha's presentation will run about 30 to 35 minutes, after which we will open up to the audience for a question and answer session. However, you can always continuously add your questions right to the end by entering your questions on the dashboard on the right side of your screen. So I want to appreciate everyone's time. Thank you, and now we'll hand it to our speaker, Sasha. Please enjoy.
Thank you very much, Vincent. So my name is Sasha Vlasov. I am a scientist from Life Technologies that is part of Thermo Fisher now. And today I'll be giving the last presentation in the series, primarily focusing on the commercialization aspects for exosomes. Exosomes are tiny vesicles, 30 to 150 nanometers in diameter, with a very sophisticated RNA and protein cargo. These vesicles are secreted by all cells in culture and in viva, and they're found in fluids in surprisingly large numbers. So each microliter of blood, serum, or urine contains millions to billions of these vesicles. And exosomes have very many important functions, which are only partially understood at the moment. Depending on the cell or tissue of origin, many different roles and functions have been attributed to exosomes. For example, eradication of obsolete molecules, facilitators of the immune response. Exosomes also have roles in antigen presentation, programmed cell death, angiogenesis, inflammation, coagulation, dissemination of oncogenes from tumor cells, spread of pathogens such as purulents and viruses from one cell to another. And very importantly, exosomes are delivering macromolecular messages, primarily RNA and protein, enabling cell-to-cell communication and signaling. One thing to keep in mind is that exosome analysis is very challenging due to their sophisticated composition and extremely small size. As you can see from this slide, average exosome contains a number of very diverse RNA molecules and protein molecules encapsulated in lipid bilayer. And also there's a good number of transmembrane proteins partially exposed on the surface of these vesicles. Each cell is secreting several types of exosomes and other nano and micro vesicles with different cargo and different functions. But overall, it's very hard to separately recover them because they are very, very similar to each other in terms of size and their surface protein signature. When you're dealing with body fluid, situation is becoming increasingly complex because all body fluids contain exosomes secreted by different cells. And as a consequence, you're supposed to analyze this very diverse and complex population that has hundreds of different vesicles in there. And they're so small that you cannot see them in the regular microscope. You're supposed to use NanoSight and other advanced instruments to tease them out. As a consequence, because of all this complexity, still there's no rock-solid nomenclature for exosomes and other extracellular vesicles, and it will take a while to get this level of complete understanding. Why we're also excited about exosomes? First of all, fundamental research aspects are amazing, and it will take many, many more years to have good understanding of the mechanism of formation of exosomes, how cell decides what needs to be packaged there, how they're secreted in the extracellular space, how they travel from one cell to another, how they are absorbed by the recipient cells, and the cargo is released, and how exosomes display their biological functions. Jan gave a great talk about this portion. Then second aspect is diagnostics and biomarkers. There's a good number of companies exploring this. Johan talked about that last week. And I wanted to highlight that there are several smaller companies that solely focus on development of exosome diagnostics, but at the same time, all big pharma such as Pfizer, Merck, Takeda, J&J have divisions also working on development of exosome-based diagnostics and biomarkers. And finally, there are very few companies that aim even higher, and they explore therapeutic potential of exosomes, which is clearly a more remote goal, but I think exosomes have really high chance to succeed in this space. So these are the list of 4 companies that primarily focus on exosome diagnostics. So you all know exosome diagnostics. Johan gave a talk last week. Caris Life Sciences, Exosome Sciences, and Exosomic Siena, which is a part of Hansa Biomed. Each one of these companies has unique technology allowing isolation of exosomes from the body fluid, primarily serum, plasma, or urine. And then after isolation step, they recover RNA and protein cargo and analyze it. So with the objective to develop diagnostic tests for cancer and other diseases. So overall, it's really amazing progress that these companies made lately. And it's interesting to see that there are newcomers. For example, there's a startup, Nanosomics. This team isolates neuronal exosomes from patient blood, and then they measure levels of Alzheimer's protein biomarkers. I sincerely hope that there will be big news towards the end of this year, maybe early 2015, about the initial diagnostic tests released, and most likely to be exosome diagnostics. What's happening today, all of these companies, they analyze their entire exosomal population derived from body fluid. But pretty much everyone's dream is to identify exosomal surface markers that will indicate organ, tissue, or cell of origin, or tumor. Once you identify these unique markers, you can develop antibodies or aptamers or some other capturing moieties that will allow to separately recover these populations. And it will be next level of diagnostic test when you start from a urine sample or blood serum and separately recover liver exosomes, brain exosomes, lung exosomes. And then each one of these fractions can be analyzed for their RNA and protein cargo and you can have very clear understanding of what's going on in this particular organ. Because in the current situation, when you analyze total exosome pool derived from blood, certain signals can be easily lost in the background. And speaking about therapeutic potential of exosomes, these fascinating vesicles are natural delivery vehicle that enable cell-to-cell communication signaling, and clearly they have huge potential as vesicles for delivery of therapeutic siRNA or microRNA or even small molecule drugs. This is one of the first papers that was published in Nature Biotech 2011 by Dr. Wood's group. In this particular setting, scientists loaded exosomes with a synthetic siRNA using electroporation, attached targeting moieties, administered exosomes IV systemically to mice, and they accomplished pretty impressive knockdown in mouse brain, both at the mRNA level and protein level. So I'm really excited about these studies. Several other groups explore that as well. But I would say I'm cautiously optimistic because I spent many years of my life developing reagents for in vivo delivery of antisense oligonucleotides and siRNA. And this field is pretty old. Overall, it's about 15 or 20 years old. Many technologies were explored, including cationic lipids, different polymers, conjugates, sophisticated nanoparticles of all kinds. And in vivo delivery is very, very challenging. Exosomes clearly have tremendous potential because it's a very sophisticated particle. It's not supposed to be toxic or immunogenic in humans, but there are a number of problems associated with them. For example, loading of exosomes with the siRNase is very, very challenging. It's not a straightforward procedure. Then large-scale production is a problem, and stability, of course, is another challenge. So the bottom line is that I think that exosomes have a really good chance to succeed in this space, but we are talking about 5 to 10 years from now. Ethlo Medical has a completely different approach using exosomes towards clinical applications. This company developed Ethlon Hemopurifier, which is essentially a canister that is connected to patient's bloodstream, and it very efficiently captures and eliminates pathogenic exosomes from patient circulation using lectins. The company completed initial early clinical trials in India, and now they plan to initiate clinical trials in the United States next month, going after cancer and hepatitis C virus. It's very promising, very unusual technology, and I sincerely hope it will work great. One company is using exosomes for cosmetic purposes. So what they do, they grow stem cells, isolate exosomes, and add them to creams and the skin serum, and these products are supposed to make ladies even more beautiful. That's a very interesting application, completely different development, and I want to see more examples like that. In the previous slides, I was talking about exosomes derived from mammalian cells, but there were several reports published over the last couple of years that clearly demonstrate that exosomes are ubiquitous, and they're found in many other kingdoms and different settings. For example, there was a paper published early this year in Science where authors discovered exosomes in seawater, and it turns out that these exosomes are secreted there by numerically dominant cyanobacterium. And the paper highlighted on this slide is dealing with grape-derived exosomes. So it turns out that exosomes or their brothers, some other vesicles, are found in plants in very high numbers. And what these authors did, they isolated exosomes from grape juice, very nicely characterized them, and then they used them in mice. And it turns out that these exosomes have therapeutic potential, and they protected mice from DSS-induced colitis. So now this group is moving forward with clinical trials where they use grape juice-derived exosomes to treat actual human patients from colitis. It's very interesting. So I'm wondering overall what's next. Grape juice is a great starting point, but what about orange juice or apple juice or bananas? Maybe they also have interesting exosomes that will be even better to your health. I'm actually not kidding. I think it's definitely worth exploring. The societies are very curious about that. Then milk clearly has substantial levels of exosomes and other nano and microvesicles. Clearly, some of them are depleted in the process of manufacturing of milk when you're moving forward with high temperature treatment or filtration, but some of them are still in milk. Yogurt, same deal. And so maybe you can envision a few years from now there'll be exosome enriched yogurt or exosome depleted yogurt. It'll be really interesting to explore. I don't know what will happen. So maybe these exosomes will be beneficial to human health, or some people will be exosome intolerant. And my favorite subject is beer. So clearly beer should have plenty of vesicles of all kinds. And we discussed it many, many times, but we never had enough time and manpower to embark on this adventure. So, but every time we had the Friday happy hour, we talked about that for the last 3 years. And now it's too late to explore that, because during the last ICEF meeting at Rotterdam, I saw 3 posters where scientists isolated exosomes from unfiltered beer, also very nicely characterized them, and they even studied body distribution in mice. Now I want to see what is actually a new development to this story. I sincerely hope that they will study effects of these exosomes in the animal models and eventually in humans, and I think these effects should be beneficial. Otherwise, I'll quit drinking beer. So I described the state of the art today, so where we are right now, that exosomes are really cool entities, and we know some things about them, but overall we don't really know the whole lot, and there's a lot to be discovered. And then there's bright future and tremendous potential where we can be with the exosomes, let's say 5-10 years from now. To close this gap, to get to the bright future, scientists clearly need tools that allow isolation of exosomes and advanced analysis of these vesicles. And that's exactly what we started working on about 3 or 4 years ago. We built a collection of kits, reagents, and instruments allowing recovery and subsequent analysis of exosomes. If you go to our webpage, you can see a detailed description of these reagents, but also, very importantly, there's plenty of very useful information. For example, documentary miniseries Exosomes, the Next Small Thing, where we have interviews from many key opinion leaders and their take on exosomes, their vision of the future. Many recorded talks from the conferences, papers highlighting recent exosome developments. There's exosome talk where you can post questions related to exosomes, so you can help other people with their research. Video Q&A, some interesting facts about exosomes. It's very, very densely loaded webpage. Next slide shows the standard workflow that we developed for isolation of exosomes. You can start from any body fluid of interest of human or animal origin or cell culture media. Isolation of exosomes can be performed using total exosome isolation reagents. There are 5 of them developed for each body fluid individually, and I'll talk about that in a minute. Once exosomes are recovered, you can purify exosomal RNA and protein cargo with a kit specifically designed for this purpose, and the downstream analysis can be done using versatile instrumentation available from LifeTech or Thermo Fisher. So on the RNA side, you can do qPCR with the Tachman assays. Sequencing can be performed either on solid platform or using one of the ion tolerant instruments. Western blotting, there are many instrument options for that if you want to primarily focus on the protein side of the things. Now I'll spend a little bit more time talking about each step within this workflow. For isolation of exosomes, there are 5 reagents. There's cell culture media reagent, blood serum, blood plasma, urine, and a so-called reagent for other body fluids that has protocols for milk, saliva, acidic fluid, and amniotic fluid. All of these reagents share the same core component, but they are carefully optimized for the specific sample type in terms of concentration, pH, salt, etc. The way the core component works is it ties up water molecules and forces larger vesicles out of solution so that exosomes pretty rapidly precipitate. For example, if you take serum, mix it with reagent, precipitation takes place within the next 15 minutes, and then you can move forward with very quick centrifugation step, pellet exosomes, discard supernatant, And the pellet can be suspended in any buffer of your choice, for example, PBS. And then exosomes are ready for any type of downstream analysis. It could be endpoint analysis such as qPCR, or it could be biological studies to find out about their networks and pathways. So to highlight the part about these kits, they enable maximal recovery of fully intact exosomes for any type of downstream assays. Protocols are very easy and reliable. There's no ultracentrifugation involved, and the reagents are very flexible in terms of sample volume input. You can isolate exosomes from 10 microliters of serum or 10 milliliters. You can process many samples at a time. It gives you much more flexibility and different options versus ultracentrifugation. The typical way of exosome analysis is using NanoSight. This instrument allows you to obtain very detailed information about the size distribution of exosomes and also concentration. So this slide shows analysis of exosomes derived from Hela cell culture media and human blood serum. And we compared side by side reagent protocol with ultracentrifugation. That is still pretty popular option for recovery of vesicles. The x-axis is the size of particles in nanometer, and the y-axis is concentration. So as you can see, with both protocols, we recover about same number of exosomes. Reagent actually recovers somewhat more. Size distribution is very similar, but in terms of duration of the workflow, a reagent is clearly advantageous because from serum it takes only about 1 hour total time to recover exosomes, and ultracentrifugation can take anywhere between 5 hours and 3 hours, depending on how you do it. With sucrose gradient, it's extremely painful and lengthy procedure. Next slide shows Western blotting for CD63, which is not a perfect exosomal marker, but it's the best option at the moment and the most popular option accepted by exosome community. So again, Hela cell culture media samples, human blood serum samples, exosomes were isolated in triplicates using ultracentrifugation protocol and reagent protocol. And in both cases, we recover very similar amount of exosomes and of very similar quality. Western blotting and NanoSight analysis is a routine type of analysis that gives you very valuable information about exosomes. But for selected samples, we perform electron microscopy, which is definitely a small laborious procedure, very tricky, low throughput, but it allows you to obtain extremely useful information about vesicles. You can take a very close look at them and very accurately determine the size, shape, and in case you do immunolabeling, you can also confirm that these entities are positive for certain markers. And that's exactly what's shown on this slide. We isolated exosomes from 2 different cell lines and performed electron microscopy analysis with the anti-CD81 and anti-CD63 antibodies. So you can see that these gray spheres are exosomes in the expected size range and shape, and the tiny black dots are gold particles that are about 10 nanometers in size, indicating that exosomes are positive for these well-known tetraspalin markers. Now, once you recover and characterize exosomes, next step is isolation of their content. We developed a total exosome RNA and protein isolation kit that is based on parental Nirvana Paris platform, but it's fine-tuned for exosomes. This kit enables extraction of highly pure RNA, including small RNA fraction down to 10 nucleotides. It allows recovery of protein from the same sample as well. And also this kit is compatible with any exosome isolation protocol. So if you choose to use ultrafiltration or ultracentrifugation or something else, you can still recover RNA using this kit. The protocol is very simple. You start from denaturation of sample, then perform acid phenol chloroform extraction, and the next step is purification on glass fiber filters. And you can either isolate total RNA pool, or if some projects require recovery of small RNA fraction, you can isolate RNA pool that is shorter than 200 nucleotides. Protein fraction can be collected at the initial processing step as well. Okay, once RNA is recovered, we routinely perform bioanalyzer and qPCR downstream assays. This is the standard profile that we see with the exosomes. As is obvious from this slide, vast majority of RNA contained within exosomes are short. Usually there's a size range from 20 to 200 nucleotides. And this includes, first of all, fragments of mRNA and ribosomal RNA, but also some full-length molecules such as tRNA, microRNA, and many other non-coding RNA sequences. But at the same time, I wanted to highlight that some exosomes contain full-length mRNA or ribosomal RNA that could be up to 5 kb long, or even longer in some cases. So these are very diverse population of vesicles, and they contain not only debris, but full-length, fully functional molecules. Tachman assays, qPCR overall is a very popular approach, of course, to analyze exosomal RNA. And in the early days of exosome research, we screened number of publications and identified number of RNAs that everyone agreed are typical RNA cargo of exosomes. And this includes LED7E, microRNA 26A, 60, and 24. Number of papers reported that they typically present in exosomes derived from different sources. And as initial QC procedure, we usually do assays with this Tachman. Again, it's Hela cell culture media, human blood serum. When you compare reagent and ultracentrifugation protocols, in both cases, we recover substantial amount of vesicles. We recover substantial amount of RNA, as can be seen from CT values in the range 20 to 35. But with reagent, we always recover somewhat more RNA, as you can see from lower CT values. But just keep in mind that the overall profiles are very similar for both isolation protocols. Now, qPCR analysis is great in case you know what you're going after. If you want to identify new sequences residing within exosomes, sequencing is, of course, the way to go. And it used to be very painful procedure until recently. When we just started working with exosomes, we used primarily solid platform, and it's amazing instrument, but overall the workflows were extremely painful, and it took a while to sequence the sample, took a while to make sense of the data obtained within this experiment. And then we switched to ion torrent platform, first PGM, then Proton, and now life is substantially easier, because construction of library 6 hours, template preparation 4 hours, running sequencing just 2 hours. And data analysis, well, it can be 1 hour if you do primitive analysis, and it can be anywhere between probably 5 and 10 hours for advanced analysis, depending on what you do. Our bioinformatics team put together a very simple and straightforward workflow. You start from raw sequence, then do pre-processing, mapping, quantification step, and at the end you have a very easy to understand Excel summary where all the sequences are listed and you can find counts next to them. Next slide shows one of our earlier sequencing experiments. In this case, we isolated exosomes from Hela cell culture media and compared exosomal RNA content to parental cells. So we are comparing them to Hela cells that actually secreted these vesicles. Experiment was done in triplicate, and the left graph shows percent mapping to RNA species for total sample, and the right graph is zooming onto the initial 0 to 10% range to view less prominent species. What is very obvious from this slide is that exosomes contain very diverse RNA cargo. Until recently, everyone was primarily focusing on microRNAs residing within exosomes and mRNA, but these vesicles contain substantial amount of ribosomal RNA, tRNA, many other non-coding RNA. And another myth until recently was that exosomes are very special and cell packages very selective species of RNA within exosomes. In reality, most of RNA found in cells is incorporated in exosomes. Of course, in some cases, levels of RNA are exactly the same in cells and exosomes. And it's great for molecular diagnostic applications because you're talking about cell biopsy. But some RNAs are found at completely different levels in cells and in exosome populations. So some RNAs we saw are 10-fold enriched, and some of them are like 20-fold depleted in exosomes. So they have potential to become exosome markers. Of course, it's a very interesting situation, very early and exciting days of exosome science. We don't have understanding how cell decide what needs to be packaged there. And also keep in mind that here and in all previous experiments, we are dealing with the entire exosomal population. So we select the total pool of exosomes and analyze them. And the way we want to be a few years from now, it will be great to somehow separately recover exosomes with a different cargo and different functions and study them one at a time. It will be much more informative this way. Unfortunately, at the moment, we don't know these markers yet because exosomes are very complex entities and they have many different proteins exposed on the surface. And if you just capture one population, let's say positive for Alex or CD63, many exosomes have these markers, and you don't really know how to separately recover these populations. Maybe it could be a combination of markers, or we can explore something more sophisticated. So what else is in the toolbox? If you want to isolate subpopulations with known markers that are not perfect, but at least they're partially characterized today, our colleagues from DNL keep expanding their collection. Currently available are DynaBeads with the anti-human CD63, CD81, CD9 AbCam. And these beads enable isolation of exosomal subpopulations from cell culture media and some other samples. If you have your favorite antibody that is missing from this collection, for example, Alex, Annexin, or something else, you can take this antibody in the bioattenuated form, mix it with streptavidin DynaBeads, and within a few minutes you have ready to go custom DynaBead that can be used for your studies. The workflows are very simple. Overall, you start with an exosome population that is pre-enriched using ultracentrifugation or total exosome isolation reagents. Then you can capture specific subpopulations with the DynaBeads, and downstream analysis can be of any kind. Of course, you can do flow analysis, electron microscopy, qPCR, Western blotting. This is one of my favorite images that shows the efficient capture of exosomes on the surface of DynaBead. Left side is DynaBead by itself, negative control without any attached antibodies, and as a consequence, no exosomes were captured there. So it's a very clean surface. And keep in mind that DynaBeads are pretty big. They're several micron in diameter. Graph on the right, figure on the right shows DynaBead with conjugated CD63 antibodies. And there's a substantial number of exosomes captured on the surface of this DynaBead. So all these tiny little gray dots are exosomes captured using CD63 antibody. It's very impressive. Now moving forward to tracing. It is extremely important to take a closer look at exosomes and analyze how these entities are secreted by cells, how they travel through extracellular space, and how they get internalized by recipient cells. There are a couple of approaches how you can do it. The most popular one is using fluorescent dyes, but if you want to have more sensitive way of enabling quantification, you can use Stackman assays and unique markers. So, but speaking about dyes, we screened a large number of fluorescent dyes from molecular probes and identified 2 leads for labeling exosomal RNA cargo. Site RNA select is a really good option. And if one wants to label exosomal membrane components, body PTR ceramide is doing a really good job. This figure shows an example for RNA labeling. So what we did, exosomes were labeled with site RNA select and added to the recipient cells. And then at different time points, we analyzed uptake and distribution of exosomes. Left figures are negative controls. The top is no treatment, and the bottom one is dye only. So clearly, dye by itself is capable of crossing the cell membrane, and it labels some RNA found in cytoplasm. But the figure on the right shows uptake of dye-labeled exosomes. So these bright green dots that you see in cytoplasm are clusters of exosomes that were taken up by cell, and they're stuck in endosomes, and their content is gradually released in the cytoplasm. When we took images at different time points, 1, 2, 3, 4 hours, we see that RNA is escaping this compartment is evenly distributed in the cytoplasm. In this current experiment, magnification was pretty low, it was only 20x, so we cannot clearly see here individual exosomes, but we can see clusters of these vesicles stuck together. So it's very impressive, and is just another tool that can be used by exosome enthusiasts for advanced studies. So to summarize, exosomes are fascinating vesicles. They're one of my favorite objects of study. I'm really excited about this area of research that is growing exponentially in the last 4 years. We already know a lot about them, and we gain more knowledge every day. But clearly, there's still a lot to be discovered. So I'm pretty sure there'll be more announcements about their functions, networks, and pathways in the next few years. And certain papers will be confirmed. Some of them will turn out to be not real or not widely applicable to different systems. But it's really, really exciting. And it's just probably the beginning of the journey. Another thing that makes me happy is that finally people know what are these exosomes. 4 years ago, when I started working with exosomes, and I was talking to a number of people at AACR and many other meetings, every time I approached a person and asked them about exosomes, typical answer was, exo what? People had no clue about the existence of these vesicles, and everyone did a really good job at promoting these vesicles and building awareness. So now we are ready for this big adventure. So to summarize, exosomes are unique vesicles with wide variety of functions, which we're only beginning to understand. Exosomes have tremendous potential as next-generation non-invasive diagnostics, biomarkers, and therapeutics. There are many other potential applications and commercial opportunities for exosomes and extracellular vesicles in addition to clinical use. I'm talking about cosmetic industry, as I highlighted in one slide. They can be used in the food industry, and there are many, many other applications out there. We launched a set of reagents enabling isolation and analysis of exosomes. But this is just the first step. Continuous development of advanced tools is crucial to further our understanding of exosomes and expanding the range of their practical applications. I'm really, really excited about this journey, and I'm pretty sure it will be many more years of excitement ahead of us. Thank you very much for your time, and now I'm ready to take questions.
Thanks, Sasha, so much for really very informative and outstanding presentation, and the various different aspects of the pipeline in terms of what you put through for the field, and also other areas like theater and cosmetics, and where exosomes play a role in. So right now we're going to continue to accept questions for those that are remaining on the line. Please use the dashboard, and even though if there's not enough time for Sasha to answer them live today, we will also provide a link later in reference to those written responses to the questions that are posted. So please continue to submit your questions. Joanne, I'm not. I don't see the question. My question box is not active, so perhaps you might be able to see the questions.
Hi there. Yes, I can do that. Let's see. Well, the first comment was whoever is sneezing. I think that was you, Winston. I took care of that.
Yeah, I apologize.
The first question is, have exosomes been used to introduce CRISPR-Cas cassettes into cells?
Oh, great question. I didn't see publications on this topic yet, but maybe I'm ignorant. Maybe some of them were published, because I know that at least a couple of labs were exploring that. So they just started exploring, but I don't think they released this data or published it in any form yet. It's a very good idea, very interesting. Because the thing is about exosomes, the beauty of them, they have pretty substantial internal volume, and they can be loaded with any cargo. It could be siRNA, therapeutic one, could be microRNA mimics, inhibitors, could be DNA, could be protein, could be anything. And I'm pretty sure a few years from now there'll be many, many examples of diverse applications of exosomes for for treating human patients or as tools.
Great. That was a question from Sam Consummate, who is one of our Scientific Advisory Board members. From Bhavani Stout, the question is, can you evaluate exosome by confocal microscopy?
Should be doable. Just overall, exosomes are pretty small in size range. They're 30 to 150 nanometers, so you can see them at highest magnification. So just with confocal microscopy, you should be able to see them, but barely. Basically, that's my understanding. We never had a chance to do it in the company, but my understanding should be doable. Our routine way of analysis, NanoSight, is already mentioned, that works great for size range. Probably between 10 nanometers or 30 nanometers to 1,000 to 2,000 nanometers. There are two other options for instruments from Izon that is based on nanopore analysis versus NanoSight is based on light scattering. And there's another instrument available from a small company based in Germany. I think it was just released.
Great. We have a question from Tom Fair. Oh, I'm sorry. The first question was from Tom Fair. And then here's a question from Sam Consummate. Are exosomes found in serum used in cell culture? If so, how do you differentiate between serum-derived exosomes versus exosomes released by the cells in culture?
Oh, yeah, that's a good point. In most of the cases when scientists grow cells and they want to study exosomes secreted by cells in cell culture, they use fetal bovine serum or FBS or calf serum as additive at 5 or 10%. And obviously, when you add fetal bovine serum to cell culture media, you introduce huge levels of foreign exosomes, because serum usually has about 100-fold more exosomes compared to cell culture media. So it's deadly. It will completely ruin your experiment. You'll be studying unrelated background exosomes. So there are two approaches that can be used to ensure that you study exosomes that were truly secreted in media by cells. First approach is use exosome depleted fetal bovine serum. So you can take FBS and remove exosomes either by precipitation or ultrafiltration or ultracentrifugation. And then you can add it to cell culture. Another approach, in very rare cases, you can find chemically defined media. There's purely synthetic, there's no FBS there. To my knowledge, at the moment, most of the people use home brew, so they prepare exosome depleted FBS by themselves. But there's a commercially available product from System Biosciences. They launched it about probably 6 to 12 months ago. And our partners from GIPCA plan to launch exosome depleted FBS very soon, in about a month.
Great. Here's another question from Sam Consummate. Can you please talk more about using exosomes to follow CNS parameters / biomarkers? And thanks for mentioning nanosomics, he says.
Just overall, exosomes are found in all body fluids. Clearly, the most popular sample types are serum and urine at the moment in the diagnostic space, but they're also found in the cerebrospinal fluid, many other samples at lower numbers, but still it's extremely valuable. The reason why everyone is focusing on serum and urine is that of course, the great value is to have non-invasive or minimally invasive diagnostics. Another reason is that blood is extremely popular in the clinical and diagnostic space, and the urine is also becoming pretty popular for different types of tests. And also, primary target is prostate cancer, and urine is easily available in this case, and it makes sense to analyze it. CSF and other body fluids are becoming gradually increasingly popular, and I think there's a bright future there. I cannot really comment more about the diagnostic side anymore, because my primary focus is on the researchers-only space, but as I see more and more companies kind of expand their scope and isolate and analyze exosomes from more different clinically relevant samples, and by analyzing their RNA and protein content, they develop diagnostics for Alzheimer, brain injury, and many other brain disorders, and as well as cancers.
Thank you. Excuse me. Alan Ezrin has a question. What are current limitations of using NanoSight system for quantitation of particles if one does not confirm that these are lipid vesicles?
That's a great question, Alan. NanoSight clearly has some limitations. It's at the moment it's the most popular instrument option. It provides really useful information, but it's not perfect. Because without fluorescent labeling, without using certain antibodies and other markers, all you can see is just nanoparticles, and you're not sure whether these are exosomes or other particles. Could be of protein nature, some aggregates in the same size range. One of the easiest way to discriminate between exosomes and, for example, protein aggregates, you can perform proteinase K treatment. And we've done a number of these experiments. You can take proteinase K, and what this enzyme does, it destroys very rapidly at room temperature pretty much the entire protein in your sample. Then it self-destructs, it chews up itself. At the end, all we have remaining is lipid vesicles, exosomes and their brothers, similar nanoparticles and microvesicles. So if you do, let's say, analysis of your sample before proteinase K treatment and after proteinase K treatment, you can see, you can determine by NanoSight whether you were dealing with true exosome samples or part of these vesicles were protein contaminants.
Terrific. These are great questions. Travis Antis has a question. Why would you suggest using ALEX on dynabeads to capture exosome when ALEX is believed to be internal to the microvesicle?
That's a great question, Travis. At the moment, there's a lot of confusion in the exosome community about the true exosomal markers. And there's a good number of papers that use this marker. I cannot really comment on that. Time will show, I guess, which ones are the best markers for exosomes. At the moment, the most popular ones, of course, are tetraspanins. It's primarily CD63, CD9, CD81. But more and more people explore other markers, and the answer could be in combination of these ones. So that's why one of our earliest product launches was actually dynabead decorated with the streptavidin, so that scientists can use their own antibody of choice, biotinylated one, whatever it is, and they can make their own magnetic particles for capture and analysis of exosomes.
Thank you. I hope I'm seeing this properly. It's Hang Wang is asking, can exosomes isolated with Life Technologies kits be used for RNA isolation with other companies' kits? Some of them seem to be easier than Life Tech's RNA isolation kits since no organic reagent is involved.
That's another great question. Overall, when you think about exosomes, there's nothing really special and magical about them. They are not more resistant to lysis. They have exactly the same membrane composition as parental cell. So the bottom line is that pretty much any kit for isolation of RNA that is routinely used on cells will work on exosomes as well. So you can use Trizol, Birvana Paris, our kits, some kits from QIAGEN and other companies. So the only unique thing about exosomes is that they are usually present in relatively small amounts. The RNA content encapsulated with exosomes is really, really low. Just to give you an idea, or when we performed calculations, it turns out that on average each exosome contains just in the range 1 to 10 RNA molecules, sometimes less than that. Of course, when you're dealing with huge population, still the numbers are large, but compared to cells or processing chunks of tissue, you're dealing with very small amount of RNA. So whatever kit you choose for RNA extraction, just make sure that it's compatible with low sample input. Just make sure that it's not gigantic column or it's not organic extraction done on large scale, because you can use your precious RNA sample. And then a brief comment about organic extraction. This step is actually optional. When you isolate exosomes with a total exosome isolation reagents, usually the samples are very clean and you can skip organic extraction part. But some of the scientists do for example, ultrafiltration procedure. And in this case, the exosome prep contains pretty high protein load, and organic extraction will ensure that you get rid of these proteins and that you perform very easy and simple lysis downstream.
Beautiful. Well, we have one more question that's come in. This is from Tom Fair, who is our Director for Strategic Alliances. What are the technologies that could be used to scale up exosome production?
Great question. I know that a number of companies explore potential for exosomes on the therapeutic side, and they develop some unusual procedures for recovery of exosomes large scale that are usually based on ultracentrifugation and variation of this theme. Honestly, I think that vast majority of clinical applications and any application basically that requires exosomes on large scale, they don't really require high purity of exosomes. So clearly, ultracentrifugation is not a great option. This procedure is super sophisticated, super tricky. You lose vast majority of your exosomes on the way. You cannot process too many samples. And every single person is trying to figure out how you can produce exosomes faster and easier and more reliable way. So if you're talking about production from liters of samples or tens of liters, definitely you don't want to do ultracentrifugation. Definitely you don't want to use antibody-based capture or lectins or our total exosome isolation reagents for precipitation. I would think that the easiest way is to use some kind of filtration, because in this case, if you carefully optimize filtration process, you can ensure isolation of vesicles, let's say in the range from 30 nanometers to 500 nanometers, and this will be pretty clean exosomes and their twin brothers. I think that's, at least my personal opinion, filtration is the way to go when you want to mass produce exosomes.
Well, thank you so much. This has been a wonderful session. Winston, do you have anything you'd like to say as we start to close? We'll have to unmute.
No, I just want to say, Sasha, that was a great presentation, and thanks for taking the time for answering those questions. And so I just want to say that there's one more session left, so please don't forget to register for the last session. And again, all this material, the question and answers, as well as the presentation and the presentation will also be available on the web. And thank you very much.
Thanks, everyone.
Thank you very much, Vince and Jen. Thank you.
Thank you.